a lot of photocopied materials to write and practice on. So that please keep that handy with you. Okay, open that. There are three sets, so you can actually waste them. You can redraw them, whatever you want, you can do. Okay, but let us make this a proper actual drawing session so that things will remain. Okay, so when you open that set, you see four different pictures. Okay, the three different sets are there, and one additional is with labeling. There is one with labeling that you can keep it aside. Okay, but then there is another thing with without labeling. You can see under surface of the brain, the hemispheres, under surface of the hemispheres in one, and there is the lateral surface in one, and there is the brainstem with the cerebellum removed in one image. So you can see all those things. You can use those things to actually draw these the things that are there. Okay. But before that, I would want you to look at the one with the labeling, the image with the labeling. Okay, the image with the labeling from the front. Okay, it contains the brainstem and all this all the cranial nerve exits. Okay, can you see the brainstem with the cranial nerve exits? Yeah, just have a look. Please have a look at that and see a note where exactly the oculomotor nerve exits. See where the trochlear nerve comes from. Okay. So these two things are the midbrain exits. Okay, that is above the pons. Then see where the abducens exits from, just below the pons. Okay. So they are little away. Okay. All the nuclei that are supposed to control eye movements are not together. They are separated by a distance that is equal to the or little more than the length of the superior inferior length of the pons. So that you just keep it in mind. Okay, that is one. Now, and then you can see where all the other nerves are: the hypoglossal, the facial, these things. Okay. This talk is actually a prelude. This topic is actually a prelude to the talk that is going to come later by Dr. Francis. So he is going to tell the meanings of different lesions and how to localize those lesions. So before that, you need to know the gross anatomy of that region, how the connections go from one place to another place, so that when that topic of actual clinical problems are introduced, we know what he is talking about. Okay, fine. So you can start using your pen and paper and start doing this thing. Okay. Now all of us know, the moment we talk about eyes, okay, the name is final common pathway, but final common pathway is not just the brainstem pathway. The pathway actually starts to control the eye movement, starts from the orbit itself, from the globe itself. From the retina. I'll tell you why. All of us know what is the function of the eyes. All of us are thinking in terms of one function, that is vision. Vision is camera function. Okay. Other than camera function, what else does it do? It takes care in balance by assessing the movement of the visual field or the world in relation to us or whatever. That is one thing. Then there is one more thing. Okay. There is one more thing that it does. It also takes part in the circadian rhythm maintenance. Okay. It also takes part in circadian rhythm maintenance. Routinely, whenever we draw as undergraduate students, when we drew about the visual pathway, from the retina the pathway starts. Okay. You can start looking at that image. You can look here at the image in this corner. Okay. Down and this corner, both sides. Okay. So you can look at that image and see and draw these things. Okay. All of us have been told, okay, it starts in the retina. There is the optic nerve. There is the optic chiasma. Okay. Then there is the optic tract. Optic tract, and then from there, lateral geniculate body, that is the thalamic radiation, and from there to the occipital striate cortex. Okay, that is the primary visual cortex. That is all we have been told. Okay, that is it. Okay, that also takes part in controlling the movement of the eye, and there is one more pathway which takes part in the controlling the movements of the eye. When you look at the retina itself, retina ganglion cells, there are two types of ganglion cells. There are something called magnocellular or big ganglion cells and parvo or small ganglion cells. Small ones, okay, synapse with very lesser or fewer, 1, 2, these kind of sensors. Whereas this magnocellular layer, it synapses with many sensors, okay, many rods and cones or whatever, many. So why should one ganglion cell, if it does synapse with so many other sensor cells, how does it serve a purpose? Because this is important in assessing movement, assessing movements of patterns. Okay, so that is why. So assessment of movement actually starts at the retinal layer itself. Okay, that anatomical correlate is there in the retina itself. Now, take your under surface diagram there and draw this. So when you see there, okay, the optic, it ends at the optic tract, probably that diagram that is there. Okay, so optic tract, just draw something, it need not be perfect. So the optic tract goes there. Okay, so you can see that it takes one route, okay, one route that goes laterally, okay, to the occipital cortex. You see that? To the occipital cortex. And then it takes one more branch that to the occipital cortex, it goes through the thalamus. The lateral geniculate body, okay, is a part of the thalamus. So it goes to the thalamus. Okay, through the thalamus it goes this. So this takes part in two jobs. Okay, one is what it is that we are seeing, and then two is where is the thing that that is of interest. That we will come to in a little later image. Then you can see that there is one more branch that will just go and land in the superior colliculus and the pretectal area. Okay, that is better seen in this image here, but just mark them and note that it takes two branches. The superior colliculus here is involved in detection of pattern movement. Okay, if large patterns of movement, that is what happens in optokinetic reflex. Okay. Now I was talking about, this is about what the pathway that goes to the cortex, the pathway that goes to the cortex. This one again, though it runs as one pathway, but then after it goes inside and almost anatomically one area, but then different columns take care of different interpretation. One takes care of color, shape, okay, those things. That is, what is it? Okay, what is it that we are seeing? Then another takes care of spatial localization of what we see. Where exactly is this? So there are two pathways there. This is a schematic of that pathway. You can take the lateral side diagram of the thing and just draw these things there. So here you see, okay, there was the primary visual cortex on the medial side, which is not there in the diagram that is given to you. So, okay, you would forget about that. Then after, this is this striate cortex. It is called striate cortex because the axons that are myelinated axons that come from the thalamus and reach here, they make, during staining, they give a striate pattern. Okay, then the secondary visual areas are not striated. So primary visual area is striated. Whenever we read books, we will keep reading about striate cortex, striate cortex. Striate cortex simply means the primary visual area, that is all. Okay, primary visual cortex. Then comes the secondary areas of the visual, which do other level of processing. What is the other level of processing? One of it, what is it and where is it? So this is area V5, okay, on the lateral surface. This is one of the key regions for the where pathway, and from there it goes to the posterior parietal cortex. Where exactly is this thing of interest. Okay, and then temporal area will tell what it is. Fine. Now, why was all this important? For again decision making. Now again you can draw this. This was the area of the where location. Okay. So the where location, okay, from the where location, we have simplified this a little, but it is okay. Okay, from the where location, one connection reaches the superior colliculus. Okay. Then one more goes to the frontal eye field. Okay, there is something called frontal eye field. Unlike the rest of the skeletal muscles in the human body whose control is from the primary motor cortex, that is here, the eye is not controlled by the primary motor cortex. The eye is controlled by the frontal eye field. Okay. So the frontal eye field is the area that gives, so that is why even a person, if they have a stroke involving this area, the eye movements will be spared, and if there is a problem here, then there are going to be some kinds of eye movements problem, not all eye movement problem, some kind of eye movement problem, voluntary eye movement problems. So this is the frontal eye field. So the superior colliculus gets a direct feed from posterior parietal cortex and also gets one more feed from frontal eye field. There is a reason, okay, why this is. Just for an example, just imagine that there is a sentry, there is a fort, there is a sentry who is watching out on what is happening here, what is happening there, what is happening there, and all these things. The sentry notes something of interest, two things of interest, one thing here and one thing there. And the sentry tells the person with the gun, see, I am seeing something of interest. You be ready with the gun. Simultaneously, I am sending this information to the commander. If commander decides you are going to shoot and which one to shoot of these two things, you will have to execute, be ready. Okay. So when the frontal eye field makes the decision and tells this area is already ready to execute those decisions. That is one thing. Then one more thing, very reflexive eye movements, sentry, no commander information, just like that, turn right, turn left, or whatever it is, something of interest. On its own it needs to take, it need not go all the way to frontal eye field. From here itself, those movements can be triggered. All these lesions will come in a separate lecture. Just to keep things a little relevant, I am just giving this. Okay. So from here to the superior colliculus, and from the superior colliculus, it goes to the pontine nuclei. Okay, within the pontine reticular formation, there is one more labeled image which gives you an idea. You can look at it a little later, whatever, which will tell about different kinds of nuclei in the pons. Okay, so this is one. Okay, fine. Now this one goes to superior colliculus 1 from the frontal eye field. Then there is one directly coming to the paramedian pontine reticular formation. One goes to rostral interstitial nucleus of medial longitudinal fasciculus. These two things. Basically, what are these two things? For horizontal saccades, the burst generator means, yesterday we were talking about shooting a missile. The eye should start from here, go to the other side. That burst generator for the horizontal movements is the paramedian pontine reticular formation. That burst generator for up and down for the vertical movements is the rostral interstitial nucleus of medial longitudinal fasciculus. Okay. So burst generator, that is the missile shooter. Okay, that is paramedian pontine reticular form for which movement? For which movement? Horizontal movement. And rostral interstitial nucleus is for vertical movement. After it has burst, to hold it there, that integrator or whatever function, for horizontal is nucleus prepositus hypoglossi, that is somewhere located here. For vertical, there will be an interstitial nucleus of Cajal which will be above here. Okay, which is not shown. Okay, so it will be there. That is it. Okay, that is not shown, but I am just showing because it is relevant now. This is all about this. Now, this same thing, okay, cortical, from the cortical areas, these things come down, but then what exactly is happening in the brainstem? A little more clarity is here with respect to only horizontal eye movements, okay. So, this is the paramedian pontine reticular formation we saw. But paramedian pontine reticular formation itself is a burst generator, but it does not communicate with the directly with the eye muscles. So it has to work through the final common pathway, and that is the lateral, the abducens nucleus. Okay, one to the abducens nucleus and one more to the through the medial longitudinal fasciculus to the third nerve nucleus. So you saw in the beginning, the first image, the third nerve nucleus is up, abducens is here. So the two things are located here. Okay, so this one makes contact through the abducens nucleus, and the abducens nucleus in turn runs one lateral rectus this side and the medial rectus of the other side to bring about one side movement. Okay, so this much. So we started from the retina, went all the way to the cortex and came back and landed up in the extraocular muscle. Did we? We did. This is, we just did one class of eye movements. What is this class of eye movements? Saccades. Okay. All classes of eye movements don't participate in the same thing. So we'll just summarize this saccadic movement a little bit. Okay. We will send you all these presentations, so you don't have to worry. I've included some of the slides which will not be used during the presentation also in this presentation, because they'll become part of your material. Okay. Okay, fine. So when you see this, there was a superior colliculus. Okay, from the cortex it came to the superior colliculus. From the superior colliculus, the burst generator. For horizontal, the burst generator is paramedian pontine reticular formation. For vertical movements, the burst generator is rostral interstitial nucleus of medial longitudinal fasciculus. And okay, for horizontal, it goes to the abducens nucleus here, and then from there it goes to the third nerve and whatever and comes to the eye. This is oversimplified picture. It is always better to start from some simple things and then go to build more detail into it. Okay. Now coming to the pursuit control. Okay, when you see the first image, okay, when you saw this image, okay, 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 when you see this image, there are two small circles here. Can you note these circles? Okay. These also get some amount of information about how from the visual cortex as to how one particular single object or single area of interest is moving in the world. Okay. With respect to world coordinates, how it is moving, this gets information. So this is called middle temporal area, and this is called middle superior temporal area. Okay, this is called middle temporal area, and this is called middle superior temporal area. Now these things are the ones which will run. So this was running the saccadic movements. This one is going to run the pursuit movements. Okay, so different eye movement categories use different areas. Okay, and also different pathways. Now we will come back to this. When it comes to then from the cortex, it will just directly land in the pontine nuclei. Okay, it will land in the pontine nuclei. Fine. Now when it lands in the pontine nuclei, you should note that what is happening here, what it has bypassed compared to the saccade, there is no superior colliculus inward. Correct? There was no superior colliculus inward. Okay, and then some other areas of the brain were not that important, okay, like the frontal eye field, these things. Now, from here, from the pons, when it comes to saccade, it was directly from the pontine nuclei, it was ending up in the oculomotor, okay, oculomotor and abducens, whatever it is, the nuclei that move the eye. But here, that doesn't happen. Okay, when it comes to pursuit control, it goes all the way to the cerebellar flocculus. From the flocculus, it comes to the vestibular nuclei, and the vestibular nuclei will reach the oculomotor. Okay, ocular motor and abducens and whatever. The nuclei that drive the eye muscles directly. So you see this from the pontine nuclei, instead of directly ending in the nuclear that are the oculomotor and abducens and trochlear, it takes a path which goes through the cerebellar flocculus, vestibular nuclei, and then abducens. Okay, which means the vestibular nuclei form the final common pathway for not just the vestibulo-ocular reflex. The vestibular nuclei also form the final common pathway for the pursuit control. Okay, for the pursuit control also. Okay. Now coming to vergence control. Okay, this is one category of eye movements which is preserved after so many different kinds of lesions. Okay, you see the we know that the problem with the medial longitudinal fasciculus, we all know when there is a uh, when somebody tries to move the eye to one side, he is able to the abduction of the abducting eye happens, whereas the adduction of the adducting eye does not happen, because from this side abducens nucleus through the medial longitudinal fasciculus, the message does not reach the opposite side's oculomotor. Okay, but then even that person is able to do vergence. Okay, which means the eye cannot move with when it wants to look to the right, but the eye will be able to move the same kind of movement the eye will be able to do when you want to do vergence. Why is this? Because the oculomotor nucleus itself is normal, but the command to look to the right, which has to reach from the abducens, is not reaching. But the command to converge when we are looking near comes from a different area. So that is what is shown here. Okay. Again, we saw the parietal occipital cortex. From there, it goes to the frontal eye field. So it is where basically in the depth plane, where is it? The where pathway we were talking about. Okay. Then from there, it goes through the nucleus, again pontine nuclei. From there, it goes to the cerebellum. Okay, from the cortex to the deep cerebellar nuclei. So from the pons, it goes to the cerebellum, and from there supraoculomotor areas, and then it goes. So what it does, it comes to the brainstem, the pons area, goes to the cerebellum, and then comes back again to the higher brainstem, and then from there it runs the. Okay, so this is a totally different path. Okay, though it is, and this is not the path that uses flocculus. It uses the deep cerebellar nuclei and the cerebellar cortex. So the pursuit path is different, this path is different. We can see which category of eye movement is affected, which category of eye movement is not affected, and draw conclusions as to which part of the cerebellum is involved, which part of the cerebellum is not involved. Okay, by practicing and seeing and analyzing many patients. Okay. This is again what we have been since yesterday morning, what we started. What is this? Vestibulo-ocular reflex pathway. So vestibulo-ocular reflex pathway, again there is an image, you can practice on that. There is the, okay, you can use, what image has been given is not from the front. Okay, there is one front sided image also, but you can use the back side image. So basically there is this vestibular nucleus, and from the vestibular nucleus the pathway runs through the medial longitudinal fasciculus. Of course, there are multiple pathways. One is through medial longitudinal fasciculus, then there is the ventral tegmental tract, brachium conjunctivum, so many different. But what is shown here is one, medial longitudinal fasciculus, and to the oculomotor, and also to the abducens. Okay, that is it. And then from there, the pathway goes. Okay, this is one image to show when the material will be given to you, it will be helpful to know where the medial longitudinal fasciculus runs, the long track of the medial longitudinal fasciculus, which is so important in so many varieties of eye movement control. This is just to show the location of that. Okay, this is to show the oculomotor nuclei and their relationship to the burst generators. Okay, you can just see this is the third nerve nucleus, fourth nerve nucleus, and then here you have the interstitial nucleus of Cajal and rostral interstitial nucleus of medial longitudinal fasciculus. Then you have the paramedian pontine reticular formation. We have the cerebellum here. Okay. So we know that they take different pathways. Okay. This is one vestibular nucleus to the cortex. There is a reverse pathway also. And all the things that are directly or indirectly related to eye movement. There is a frontal eye field. You can mark all these things. There is a supplementary eye field. Then insular cortex, area 3A and 2V, all these things Dr. Francis will be explaining. Posterior parietal cortex, the Weir path. Okay, the V5 area, this is the secondary visual areas, whatever, or higher visual areas. This is the primary visual cortex. Okay, so some of the Weir information is processed here and then projected here. From there, frontal eye field, supplemental eye fields, and from there they start coming down. And insular cortex is related to the vestibular related assessment of body motion and all those things. Okay, so to summarize, we started with this. We started with from the retina. There is a pathway which breaks into two. Okay, it breaks into two. One pathway went to the primary visual cortex via the thalamus. Another pathway came to the superior colliculus, okay, and the pretectal areas. Okay, and then from the superior colliculus, there are connections to the brainstem nuclei, which directly, some of the nuclei for some categories of movement to saccades directly communicate with the brainstem nuclei communicate with the oculomotor. When it comes to pursuit, colliculus involvement is not there. From the cortex, okay, it comes, then it comes to the pontine nuclei. From there it goes to the flocculus. From the flocculus comes back to the vestibular nuclei and then goes into the to run the oculomotor nerves. And when it comes to vergence, flocculus part of the cerebellum is not involved. It is the cortex and the deep cerebellar nuclei that are involved. And then from there again it goes to the here. Okay. From there it goes to the brainstem nuclei and then to the here again. What you see, what is bypassed here? What is bypassed here is for vergence, the vestibular nuclei don't take part. For smooth pursuit, the final common pathway for smooth pursuit is the same as the vestibulo-ocular reflex. Okay, the vestibular nuclei is involved. From the vestibular nuclei, this thing. For saccades, vestibular nuclei is not involved. For vergence, vestibular nuclei is not involved. For vergence, flocculus is not involved. For smooth pursuit, flocculus is involved. For smooth pursuit, the rest of the cerebellar cortex and deep cerebellar nuclei is not much important. Whereas for vergence, it is important. Fine.